everybody. As promised, I'm going to show you how to use this online website on how to create primers and run digests, which really help to complete the last problem set where you worked with a sequence and had to create primers for it and run digests with particular enzymes. So to do that, I'm going to remind you what your problem set was, and then I'll walk you through each step. So first, you guys had to go online and learn about PCR and learned about how to design a PCR for this N NC102 gene that is from the yeast genome. And from there, you had to determine how the PCR reaction would work when you use a restriction enzyme called NAD1 and KPN1. So in order to do that, I want to start by showing you how we download the sequence, how we create this account online, and then we'll work with the digest. I'll remind you a little bit about the biology of DNA, and then we'll design some primers, okay? So to do that, first we want to go to the yeast genome websites. You would go right here, you would just Google it. My internet's a little bit slow, but you would click on here. And then on the top right, you would search the sequence. So once you enter that in there, you should get this to work. And if you scroll down, you're going to have this option of download, and you're going to download the genomic DNA. You can keep it in your downloads photo for now, or you can save it in a particular folder. But for the sake of time, I'm just going to show you how to do it from your downloads photo. Next, we're going to go to benchling.com and you're going to sign in. If you do not have an account, you need to create one with Gmail. It is free and this is super helpful. So I'm just going to log in with my Gmail. And as of now, I already have it up, but I'm just going to go ahead and erase it just so that I can show you guys how to make this work out. So when you open your Benchling, you're going to be able to create different projects. I suggest that the first step you do is create a folder called Bio 220. Let's just do it for now. This is going to include anything related to that project. Within the project, you're able to create folders, entries, protocols. What we want is we want to import a DNA sequence. So you scroll down here to DNA RNA sequence, and then you scroll down to import DNA RNA sequence. You click that, then you click choose file. It should be under your downloads, and it's this first one. Once that is done, make sure that upload's done. Make sure that you click on it so that it will open, and then click close. Then this pops up. So the cool thing about this is that it tells you the sequence, it tells you how long it is. So this is roughly from 0, that's your start codon, ATG, all the way down to about 522 amino acids long. It tells you where different enzymes will actually cut. So you have NDE1 right there. You have a couple other ones throughout, but we'll go through that in a second. Another cool feature from this is you're able to see it linear. You're able to see more information regarding it. But for the sake of this purpose, we're going to stick on sequence map. Now I'm going to move myself a little bit over. So on here, you should be able to see on the right-hand side different options. And there should be this little scissors right here. If you click digest, you are able to look up the enzymes that you want this sequence to be digested. So let's go back to the problem set. It's telling you it's going to go ahead and want you to use these two enzymes. We're going to work one at a time. So to do that, we're going to search NDE1, click it, make sure it's selected, run digest. So earlier I told you that this gene is 100 to 522 amino acids long. When you click digest, it tells you exactly where it cuts. So it's cutting between position 138 and 139. That in turn tells you how the gel will look, that you have these two bands. One that's a little bit smaller. So remember when you have a gel, it actually runs quicker if it's smaller in weight. If it's bigger, this is roughly 384, it's going to be above. So this is how this is able to help you. So if we go back to the problem set, it tells you draw a gel and show me how the bands will look with an uncut. Well, we know the first one is a 522 amino acids long. And for this bottom one, sample two, now you know where it cuts. You know that there's going to be a sequence that goes from 0 to 138. So that's a band at about 138. You also know that it cuts it so that there's two now, two separate um ends with this particular enzyme. So the next one is 384. So then you would draw a line at 384. And the same thing can be repeated for KPN1. So if we go back to the sequence here, click digest and then do KPN1. Oops. There, make sure it's under selected, run digest. It tells you the length as well. If you want to do the third, I believe it's the third or the fourth sample where you cut it with both, then you go back to the sequence and you're actually able to select multiple. So we already had KPN1 on there, so I'll select that and I'll select the other one, NT1, and run the digest. And now you know how big it is and where it's cutting it. And that should tell you how to draw your gel. It's really, really cool. I highly suggest that you get a Benchling. It's going to be really helpful when it comes to creating primers and also running digests. And just so that you know what to expect on your gel as well. So next thing we're going to do is primers. So I worked a little bit backwards, but the reason for that is because I want to really illustrate and remind you of how the DNA is organized. Remember, DNA is 5 prime to 3 prime, and then here it runs from 5 prime to 3 prime. So something you got to remember is that when it comes to creating primers, it's always going to run from 5 prime to 3 prime. So in the example here, the bottom strand, which is this right here, is already 3 prime to 5 prime. But then your DNA polymerase is going to come and it's going to run 5 prime to 3 prime. Because that's essentially what it's creating. Another one here on top that runs 5 prime to 3 prime. Same thing goes for this other one. And this is basically how PCR works. You have your DNA of interest. You create primers that always are going to be running 5 prime to 3 prime. Same thing for this one on top, right? So see how these black arrows are showing you it's going like that. In order for this to happen, there has to be multiple steps. First, you have to create the correct primers, and then you have to run PCR. And when you do this first cycle, you have these two. But now you want to repeat exactly what happened here on the left for these two separate ones. And eventually you're going to get multiple cycles like this, your second cycle, then your third cycle. And if you repeat it for 20 to 30 cycles, you're eventually going to get millions of copies of it. So let's see how easy it is to use Benchling to create this primer. Let's go back to Benchling, go to your sequence, and in order to do this, I want to let you know that Marcus mentioned usually these are about 20 to 25 amino acids long. So for the sake of this purpose, I believe your problem set asked for you to do 20 acids. Yes, 20 base pairs long of an oligonucleotide. So just if you don't know what this means, an example of it is the primer that we're creating. So what we're going to do is, like I mentioned before in this picture, is the first one we're going to make is a forward one. So that one's easy. All you have to do is highlight the first 20. And the cool thing about Benchling, it tells you here in the bottom how many base pairs you are in. So I'm a little bit over, let's see, there you go. I highlighted them. Now on this top right where it says create, you're going to press create forward primer. And it automatically gives it to you right here in the 5 prime to the 3 prime. Notice it's the same thing. The reason for it is not going to change. It has to run forward. What your primers are doing is they're amplifying this gene. So that means you need primers that can recognize the beginning, and you need a primer that's going to run from the end back inwards. Why do we want it to go from right to left? It's because if you did it the other direction, you wouldn't actually amplify this gene. Your primer would be running left to right. So it would be anything that's downstream, and you don't want that. So for now, we just did our forward primer, super easy. All you would do is create it. Like I said here, primer, forward. So I already created it here, so I'm just going to click here, and I would just write this down for your problem set. That's what it is. Now, for your reverse, again, you have to consider your last 20, and you have to consider your stop codon. Usually in a lab setting, we don't include them. For the sake of this, I just want to show you a very simplified version of creating primers. Make sure that if you are in a lab, you confirm this with your advisor. But for the sake of this, I'm just going to show you how to do a reverse primer using Benchling like this. So highlight the last 20, create primer, reverse. Notice that now it's backwards. It's still 5 prime to 3 prime, but see how it's TTA. Let's go back to this sequence. It's no longer, like I said, reading left to right. It's reading the bottom strand, right? Because remember, ATG above here is going 5 prime to 3 prime. So this bottom strand is going 5 prime to 3 prime, because that's what you want your primer to be. And when you design your primer, what you would have written on your problem set is this. Just for the sake of timing, this is basically a quick little summary on how to do this so that you can run PCR and get millions of copies of your DNA of interest. I hope this helps. Good luck, you guys. Bye bye.